Welcome to today's class. Today this is a third class for Biology CBSE NEET, and today we will be discussing the topic that is principles of inheritance and variation. Let's start the class now. Today's topic is principles of inheritance and variation. This is a really interesting chapter where you come to know about how as a human or as an organism, how we inherited traits or characters from our parents, how the organisms are showing the particular trait, or how they are having individual traits and how they are varying from other organisms. So the basis of this is the genetic material. What we have, it is DNA. It is a molecular level of biomolecule present in our body. So our cell is having the nucleus, which consists of the DNA, which is the genetic material. And the coding or the message or the nucleotides present on DNA, it represent or it signifies the character or the trait shown by a particular organism, and it is inherited from his or like from the organism's parents. So we'll start with the principles of inheritance and variation. So this chapter is completely about the Mendelian genetics and the variation. We'll come to know about who Mendel was and how the term genetics came. So genetics is a term which refers to the study something related to gene. And we know that gene is part of a DNA, which is a functional part which can code for a particular protein. So if this is a DNA, a particular part of DNA is able to code for a particular protein by transcription followed by translation, and that particular DNA will be named as a gene. And the study of anything related to gene is genetics. So all living organisms reproduce, and it results in the formation of offspring of the same kind. As I mentioned right now, yeah. So it results in the formation of offspring of the same kind. It means from parents, the traits are transferred or inherited from the parents to the child or the offspring. That is the children of the parents. So this resulting offspring most often do not totally resemble the parent. As we know that we are not completely similar to our parent, we'll be like somewhat similar to the parent because we have inherited some traits. Not only it is like parents, it might be like mother or father. So we might be looking like our mother, or some might be looking like their father, or some may be looking like both their mother as well as like a mixture of mother and father characteristics. So it is like the offspring it is not compulsory that they do not completely resemble the parent. So siblings sometimes look so similar to each other or sometimes even so different. So it depends upon, because we are like both mother and father will be representing or contributing certain traits to the progeny or the offspring together. And in that case, as sibling, we could have some similarity compared to the parents. And in some cases, yes, the parents may be like more similar to the kids also. It depends upon the process, what is happening during the cell division, like the segregation process, how the combination is happening during the fertilization and all. So now moving to the terms related to Mendelian genetics and variation. First term, as I told, it is genetics. So genetics is a branch of biology which deals with the inheritance as well as the variation of characters from parents to offspring. So as I mentioned, if for example, considering a particular trait that is hair color. So if a child inherited this particular trait or hair color or texture from a parent, so that trait is inherited from the parent to the child, and maybe it could it might have in the similar texture or similar color, but it might have some difference as well. That is what variation refers to. So genetics is dealing with the inheritance as well as the variation of characters from parents to offspring. Now moving to inheritance. Inheritance is a process by which the characters are passed on from parent to progeny, and it is a basis of heredity. Okay. Now what heredity is? So heredity is a transmission of characters from parents to their offspring. So it is somewhat similar to the term inheritance. So it is like if a parent is transmitting or transferring a particular trait to their progeny. So parent to progeny, that is known as inheritance. And this transmission of that process is known as inheritance, and the transmission of characters from the parents to their offspring, that particular transmission is known as hereditary. So now moving to variation, as I mentioned, it is a degree by which the progeny differ from their parents. So how much they differ from each other, that is a variation, what they have from each other. Maybe like, for example, the parent will be having a black color eyes, and the progeny might be having brown eyes instead of black eyes. So that is a variation happening there. Now, next one is environmental variation. So these are acquired and non-heritable variations. So these are not inherited variations, they are acquired variations or non-heritable variations. Now, hereditary variations, these are genetical and inheritable. So hereditary variations means they are related to the gene and they are inherited. Okay. Now we are moving to the person behind this study, all the studies, whatever that is mentioned in this chapter, that is Gregor John Mendel. So he is known as the father of genetics. So this is very important. So you might get some MCQ questions in your NEET exams. Who is the father of genetics, or what is the role of Gregor John Mendel in genetics? Okay. So he conducted hybridization experiments on garden peas for 7 years. So the experiment was done on garden peas, and he proposed the laws of inheritance in living organisms. So he used the garden peas to study the laws of inheritance, how the characters are being transmitted from the parent to the progeny. Now, during Mendel's investigation into inheritance pattern, it was for the first time that statistical analysis and mathematical logic were applied to problems in biology. So that was a new trend in terms of research or study in case of biology, where statistical analysis as well as mathematical logic were applied. Now, Mendel investigated the characters in the garden pea plant that were manifested as two opposing traits. That means he took two plants which were having two different traits. That is, one was tall and the other was dwarf. And another character was, one was yellow and one was green. So he took two plants which were having two opposing traits. He did the experiment in for one period of time. He will be taking two plants which will be having two opposite traits. Okay. Now, Mendel selected actually 14 true breeding pea plant varieties. So 14 plants were taken, and out of this 14 plants, we could say there is 7 pairs of plants. There is 7 pairs, and the 7 characters were studied in this experiment. So as pairs, which were similar except for one character with contrasting traits. So some of the contrasting traits selected were the smooth or wrinkled seeds, yellow or green seeds, inflated constricted green or yellow pods, and tall or dwarf plants. So here we have the diagram of that. So he took the stem, which is having plant height as a character and a flower position. So these are first two characters. So plant height in the sense tall and short. Now flower position, he considered the axial position and the terminal position. Now coming to taking the consideration of flower, the flower color was studied. That is not studied. He took two different colored flowers. That was purple and white. Now the pod color, that is green pod and yellow pod. About the pod shape, it was inflated and constricted, which was studied. And coming to seed color, it was yellow seed and green seed studied. Now coming to seed shape, it was taken round seeds and wrinkled seeds. So here you can see there are 14 traits, in total 14 traits were studied, 14 true breeding pea plant varieties were trained, and we could say it was 7 pair. 7 pair in the sense plant height, flower position, flower color, pod shape, seed color, and seed shape. Now, why Mendel selected the pea plant? So what was the peculiarities or the importance of or the specialities of pea plant? So pea plant, like they have pure varieties. That means they are available they are having more pure varieties and they are easy to cultivate, and the life cycle of the plants are only few months. So that means they will be able to get the results so soon. They don't have to wait for a long period of time. So the results can be got early. Then contrasting traits are observed. As we mentioned in the previous slide, we could choose 14 plants with 7 pair characters. We could get like 7 contrasting characters here. So that was an advantage. Now next was the flowers are bisexual, as well as they are normally self-pollinated. And the flowers can be cross-pollinated only manually. And the next point which was considered was the hybrids are fertile. So they are able to produce the hybrid plants were able to produce a new plant as well. So now let's move to some terminologies related to this chapter. First is phenotype. So phenotype is simply a character. So for example, considering human, the eye color, the hair texture, the skin color, the shape of our nose, or the kind of features, the skin texture, what we are having, these the characters what we have, any kind of diseases or abnormalities, whatever we are having. So these are the phenotypes. Okay, so the external appearance of an organism due to the influence of genes and environmental factors is termed as phenotype. It is simply the external appearance, or we could say the term trait. Okay, now moving to the next term, that is genotype. So genotype refers to gene, that is something related to the gene. It is genetic constitution of an individual responsible for the phenotype. That means a particular phenotype could be represented in terms of genetic constitution. That means, for example, an eye color, considering a trait eye color, this is a phenotype. So if this is caused by a particular gene, and we can represent a gene by, for example, we can represent this color by this particular trait by the letter E. So E is the gene which causes the eye color of a particular organism. Okay, so we can represent this particular gene by different letters. That means they be either dominant or recessive, and this is the genotype. So if this particular, for example, if they are having homozygous genotype, that means they'll be having 2 dominant alleles. So if they are having 1 dominant and 1 recessive allele, means they are having heterozygous genotype. Okay, and if they are having 2 recessive alleles, they means they are having homozygous recessive genotype. So these are the genotypes, and the genotypes will be presenting or causing a particular phenotype in an organism. For example, the eye color. Okay. Now coming to phenotypic ratio. So it is a correct proportion of phenotype in a population. So how much in a particular crossing over, how many progenies were having the particular trait. The phenotypic ratio represents the proportion of phenotype in a population. Now genotypic ratio is the proportion of genotype in a population. Now, as we mentioned already, the homozygous and heterozygous. So homozygous are the individuals having identical genes in an allelic pair for a character. For example, capital T, small t, that is homozygous. Both the alleles will be similar. For example, in a chromosome, so we are having the homozygous chromosome here, and considering a particular gene, we are representing a particular gene on this chromosome. So both are the gene T. Okay, maybe in this particular homozygous means in this chromatid it would be capital T, and in the other one it could be small t or capital T. Okay, so if it is capital T, that means in both the chromatids, if they are having capital T, then the individual is having a homozygous genotype. That is capital T, capital T. If it is small t, then it will be homozygous genotype. It will be a homozygous recessive genotype, because the dominant allele is capital T and the recessive allele is small t. So this will be representing a homozygous dominant genotype, whereas small t will be representing homozygous recessive genotype. Now, what if both the chromatids have different alleles? Suppose if one is having capital T and other is having small t, it is unidentical genes. Okay, so that is a heterozygous. So the individual having unidentical genes in an allelic pair for a character is termed as heterozygous. Now, moving to the next term, that is dominant gene. So dominant gene expresses its character in heterozygous condition. So for example, if it is T, capital T, small t, and the next one is the heterozygous capital T, small t. In case of homozygous dominant, both the alleles are same, and we can't say one is dominant over the other. Both are similar, so the character will be expressed whatever the allele refers to. For example, if T is referring to the tall character, the plant or the organism will be tall. If small t is referring to the dwarf character, so that means the plant will be or the organism will be dwarf. Now, in case of heterozygous condition, if T is one particular chromosome or the one particular gene is having the capital T allele, then that is dominant. So capital T is the dominant one. So that means the character will be tall. Whereas small t is recessive here, they will not be expressing. Instead of small t, the capital T allele will be expressing there. As a result, the gene that expresses its character in heterozygous condition is the dominant gene. So if an individual is having this heterozygous genotype and he is showcasing a particular trait, for example, the tall trait, then we can say this particular trait is the dominant character there. So capital T will be dominant. Now recessive, the gene that fails to express its character in heterozygous condition. So in this case, small t is failing to express its character, that is dwarfism. It is failing to express it. So that means dwarfism is the recessive character there. Now what is hybrid? So a progeny obtained by crossing two parents that differ in characters, that is called hybrid. So if two parents are having different characters, for example, this is a tall plant and this is a dwarf plant. So both are different characters. So when we cross them, we'll be getting a hybrid. Okay. Now what is backcross? So cross between F1 hybrid and one of its parent. So if this particular progeny is crossing with one of its parents, that is, it could be like either the dominant homozygous one or the homozygous recessive, it could be any one of them. So if a cross is happening between the F1 hybrid, in the sense the progeny which has happened by crossing two parents that differ in the characters. So when this F1 hybrid is crossed with another person, another organism which is having the same trait or same character of the parent, then that is back cross. Now in case of test cross, what happens is that the cross is happening between the hybrid and it is homozygous recessive parent. That means it is between the homozygous recessive one, small t, and it is used to identify the genotype of the hybrid. Suppose if we do not know the genotype of the hybrid, then we can cross the hybrid with its homozygous recessive parent and we can find out what is the genotype of the unknown hybrid. Now, moving to the inheritance of one gene. So we are focusing on one gene, in the sense one particular trait. For example, as we are using in whole the chapter is completely focusing on mental study, and we are considering the tall and dwarf trait, that is the height of the plant is being considered here. So we'll use this as actually the trait here, that is consider the height is being studied, height of the plant is being studied in this context right now. Now, inheritance of one gene can be explained by monohybrid cross. So, mono refers to one. So, one hybrid cross, in the sense they are considering one particular trait. As we discussed right now, the capital T crossing with a small t, we have a hybrid, that is capital T small t. This is a monohybrid cross. That means we are taking one particular trait is under consideration. That is the height of the plant. Now, the cross between two parents differing in one pair of the contrasting character is called the monohybrid cross. That is what I mentioned right now. The cross between two parents, that is one is homozygous dominant and one is homozygous recessive. So they are differing in one pair of contrasting character. That means one is tall and the other is dwarf. And this is called a monohybrid cross. Now, when we cross the tall and the dwarf pea plant, what will happen? We will get the first hybrid, and this is known as F1 generation or filial generation. So, in F1, we are having a heterozygous genotype. And when we observe the plant, we observe that the plants were having the trait tallness. That is, they were tall. They were having the same trait as one of their parent. It is a tall plant. So we could say T is dominant here and the small t is recessive here, as we mentioned in the previous slide. So F1 plants were tall and none of them were dwarf. Okay. Now, for other traits, the F1 generation resembled only one parent and trait of the other parent were not shown, as we discussed right now. Now, the second case was they did that they did the pollination of F1 generation with each other, self-pollination. That means F1 into F1, we could say. Or we can say it is a heterozygous genotype crossed each other. Okay. Now what happened was the progeny had all the three types. That is, they were tall, they were heterozygous genotype, they went tall again. Then they also had homozygous recessive genotype. Three types were there. Okay, so that means they had dwarf as well as tall characters, and as you can see in this diagram, as I already mentioned, the tall were crossed with dwarf in the first crossing over. We had a hybrid here. So that is capital T small t, that is heterogeneous genotype, that is a F1 generation. All were tall, all were capital T small t, that is heterozygous genotype. Now when we mated self-pollinated the F1 hybrid, we got tall, and dwarf. That means 3 tall and 1 dwarf. That is 3:1. That is phenotypic ratio. It is about the character we are discussing. So it is not about the genotype we are focusing here. We are focusing about the character. So phenotype is representing the character. So we have 3 tall and 1 dwarf. That is 3:1 ratio. Now considering the genotype, we are having one set of capital T, one set of small t, and two set of the heterozygous genotype. It is a hybrid genotype. So here we could say it is 1:2:1 ratio. Okay, isn't it? You can see here, right? 1:2:1 ratio. It is the genotype ratio. So as here it is given 1/4 were dwarf and 3/4 were tall. So F1 generation, one parent trait shown in F1 generation, all the plants were tall. So one parent trait was there. Now in F2 generation, both parent traits were shown in the ratio 3:1 and no blending were seen. That means no hybrid kind of character were seen in F2 generation. Clear? So this is about the inheritance of one gene. Now moving to the continuation of this part. This is about the Mendel's proposal or the Mendel's theory. So he proposed or he studied that, and he gave some theories that something is being passed to the next generation through gametes. That means, as you can see, when the parents were crossed in the first case, so the male and the female parents were crossed, and we got an F1 generation here. Now after that again they were crossed, so we got an F2 generation here. So we could see that the trait of one parent was showing in F1 generation, and in F2 generation we could see that both parent traits were showing off. So that means something were passed to the next generation through the gametes, that is called the factors or genes. So through gametes, in the sense, in case of mother, the egg is being formed, that is a gamete. In case of father, the sperm is produced and they fuse together to form the progeny. So something is being passed through the gametes, and what is that? And he named it as factors. So this is an important term. Remember this point, that is, he termed the factor, the thing, that the particular substance or the factor that being transmitted from one generation to another, he named it as simply factor, and later on it was named as genes. That is simply what? The genetic material, DNA. Now, the genes are factors. So they are the unit of inheritance. So why we are like we are looking similar to our parents, or why we are having the characters similar to our parents, it is because of the genes, the DNA, what we inherit from our parents. And they contain the information required to express particular trait. Now the genes which code for a pair of contrasting traits is called the alleles. As I mentioned earlier, for example, the trait we are considering the tallness of a plant, and we represented the letter T for it. That is a gene representing tallness. And we know that the gene can exist in two forms, that is a dominant form and a recessive form. So this is allele. So these are alleles. These are two types of gene. A gene can exist in two forms, that is allele. So we can say they're existing in pairs. Okay. So a gene, for example, we could say they're existing in pairs. A gene can be existing in pairs. So we represent these characters using alphabetic symbols. That is T for tall, T for small, T for dwarf, and the plant pairs of alleles for height. So capital T will be representing tallness. Capital T, small t will be representing again tallness, whereas small t will be representing dwarfness. So the plant pairs of alleles for height. Okay, this one is clear because we have already discussed in the previous slide. Now Mendel proposed the true breeding tall or dwarf plant, that is, he proposed the homozygous allele pair, that is capital T and small t genotype will be representing homozygous alleles, that is homozygous genotype, which will be coding for the particular trait, either tall or the dwarfness. Now, coming to the descriptive term, tall or dwarf, tallness or dwarfness will be representing the phenotype, the character. Okay. Now, then the next point was that he found out that the phenotype of heterozygote, that is the capital T, small t of F1 was same as the parent with capital T, and proposed in a pair of dissimilar factors, one dominates the other and hence called dominant and recessive. So overall, we could see that here Mendel put forward the presence of factors, that is genes. He represented the two types of genes, existence of alleles, the symbols used to represent the characters, then the genotype, phenotype, and the dominant character as well as the recessive character. And also genotype is both, there are homozygous as well as heterozygous. Now, coming to monohybrid cross, as we mentioned, the phenotypic ratio is 3:1 and genotypic ratio is 1:2:1. Now we are moving to the next term, that is Punnett square. So this Punnett square, as you can see, this square is representing the Punnett square, which is used to, this is a graphical representation which is used to calculate the probability of the possible genotypes in a genetic cross. So if we are crossing two hybrid of F1 hybrids, so we can find out the possibility of the progeny by drawing a Punnett square. So production of gametes and formation of zygotes can be calculated by the Punnett square. It was developed by the scientist, British scientist, and under his name it was named as Punnett square. And the gametes are written on two sides, on row, that is, you can see the gametes are written on top as well as the left columns, and they'll be crossed, and we'll be writing on the each columns, like each squares will be writing the progeny genotype. Now, as we discussed what test cross is like crossing the F1 progeny, the heterozygous progeny, with a homozygous recessive parent. Now, moving to the Mendelian law of inheritance. So, Mendel proposed three laws, that is law of dominance, law of segregation, and law of independent assortment. So what is law of dominance? So law of dominance is that when two different alleles are present, only one is dominant and will be expressed. For example, capital T, small t, this particular genotype express the trait or phenotype that is tallness. So here capital T was dominant and small t was dwarf. Okay, sorry, recessive. So now moving to law of segregation. So law of segregation says that the two alleles for each gene are placed in different gametes. For example, when we cross the F1 generation parents, that is for the F1 hybrid, we cross two different trait parents, that is tall and dwarf. So first they segregated the gametes. Actually, they segregated in the sense the alleles for each gene were placed in different gametes. Here also we have different gametes, and then these gametes fuse together. For example, if this is a mother, so the mother will be segregating the chromosomes during meiosis. What happens is that they will be segregating the chromatids. As a result, each chromosomes will be having the two different alleles. So what happened is that in coming to the progeny, they will be having both the alleles, one from mother and one from father. It is not that the progeny is having two alleles from a single parent. One is from the mother and one is from the father. That is law of segregation. Now moving to law of independent assortment, it is related to the dihybrid cross. So here it says that the inheritance of one gene doesn't affect the inheritance of any other gene. So this is related to the inheritance of two or more genes. Okay, so we'll be explaining it in later slides. Now moving to law of dominance, as I mentioned, the characters are controlled by discrete units called factors, and factors occurs in pairs. And in a dissimilar pair of factors, one member of the pair dominates the other. For example, capital T, small t, capital T is dominating the small t. So that is why it is having the character tallness. Now, law of segregation. As I mentioned, it states that when a pair of factors for a character brought together in a hybrid, they segregate during the formation of gametes. So alleles do not blend. That means both characters are recovered in F2 and one in F1 generation. So factors which is present in parents segregate and gametes receives only one of the two factors. So here you can see in this diagram, capital R, they are segregating each allele and two different gametes, and here also they are segregating two different, means the alleles to two different gametes, and then they fuse together to form the different progeny having random combinations. Not exactly random, we'll say, like this particular gamete will be fusing with R, and if that is the case, you'll be having this particular progeny. If this R is crossing with this particular gamete, then we'll be having this particular progeny in that way. As we are having this is F1 generation, we'll be having all as the heterozygous genotype here. Now coming to this case, F2 generation, here we can see when the dominant allele is crossing with the dominant allele from the, for considering if there's a mother and if there's a father. So if this dominant allele is crossing with the dominant allele, we'll be getting a homozygous dominant genotype. Now, if this this dominant allele is crossing with the recessive allele, you'll be getting a heterozygous genotype. So depending upon the segregation, the random combination happening, what will happen? We'll get different progenies having different traits. So homozygous parent, they have one kind of gamete. Heterozygous parent, they have two kind of gamete. Each one have one allele with equal proportion. Okay, moving to incomplete dominance. So incomplete dominance is, as the name says, it is showing incomplete dominance. That means none of the character or the allele will be dominant over there. For example, as we mentioned in the previous slide, in case of heterozygous genotype, capital T were dominant over small t. As a result, it's shown the tallness. Whereas coming to incomplete dominance, none of the allele will be dominant there. They'll be showing a different trait over there. So this was discovered by Correns in Mirabilis gelata, and it is also called partial dominance or semi-dominance. So here the inheritance in which the allele for a specific character is not completely dominant over other allele is called incomplete dominance. For example, when they cross the snapdragon flower, that is a red flower with a white flower, they got pink colored flower, not red one, they got the pink colored flower. And when they self-pollinated the pink colored flower, they got red, two pink, and they got one more, that is the white flower. So here we can see the genotypic ratio is same as the Mendelian cross, whereas the phenotypic ratio is different. So here R in case of heterozygous genotype, R is not dominant over small r. So if it was dominant, it would have shown the red color. But here, as it is incomplete dominance, it showed pink colored flower. Now, and also you can see as given here the genotypic ratio. So genotypic ratio is 1:2, 2:1, that is 1:2, 2:1 is correct here, similar to the Mendelian cross, whereas the phenotypic ratio was different. Here red is 1 and pink is 2 and white is 1. So it is also having 1:2, 2:1 ratio, whereas in Mendelian cross it was 3:1 ratio. Okay, now yeah, this is an example for incomplete dominance in snapdragon flower. Now moving to co-dominance. So it is a different case in case of expression of characters. So what is incomplete dominance? So incomplete dominance is where both alleles for a character are dominant and express its full character, and it is called co-dominance. And the example will be ABO blood grouping. So there are A blood group, B, AB, and O. So it is depending upon the alleles, that is I, A allele, I, B allele, and the recessive allele, that is O allele. So if a person is having A, B blood group, and the person will have IA, that is A allele, as well as B allele. Here we can see both the alleles are dominant. We can't say that IA is dominant over B because both alleles are expressing here. So IA allele will be coding for antigen A and IB will be coding for the antigen B. So in case of like AB blood group system, both A antigen and B are present. So that means they'll be having a genotype IA, IB, where both are dominant. So similar, you can see AB blood group, they're having a genotype where both are dominant. It is co-dominance. Now moving to inheritance of two genes, that is a dihybrid cross, where Mendel explained his second law, that is law of independent assortment. So the law of independent assortment states that The factors for different pairs of contrasting characters in a hybrid assorted independently during gamete formation. And the Mendel's second law can be explained by dihybrid cross. So here the cross between two parents which differs in two pairs of contrasting characters. So let's explain a dihybrid cross here. So two characters were studied. First one was a round yellow seed and this was a wrinkled green seed. And this is a two different traits are studied and the genotypes are represented here. That is capital Y, and capital R for round yellow, and for wrinkled green it is small y and small r. Now they form the gametes, and here you can see the gametes are having both the alleles, like that means this is representing the independent assortment. So here you can see that for this particular trait, that is the yellow color of the seed, they are represented by a YY genotype, and this Y is segregating or assorted separately, and the second gene is assorted separate, that is independent assortment, the law of independent assortment, where two genes are like they are transmitted, they are like separated independently, they do not affect each other. In the gametes they'll be having both the alleles together. Okay, so this is law of independent assortment. It is similar in case of the yellow green wrinkled seed also. And we got the F1 generation here, which is having the yellow round character phenotype, but the genotype is different. It is hybrid here, and it is having heterozygous genotype for both the traits or both the genes. Okay, now because of this independent assortment, when we cross the F1 generation, again they are forming gametes, that is all the different genes are segregating or independently assorted, they are assorted independently, and finally we are getting the progeny here. The phenotypic ratio is 9:3:3:1, that is 9R of round yellow, 3R of round green, 3R of wrinkled yellow, and 1 is of wrinkled green. So there is 9:3:3:1 ratio. So this is about the independent assortment. Now moving to dihybrid test cross. So it is similar to the monohybrid test cross. Here the F1 hybrid is crossed with the recessive green wrinkled pea plant, and the phenotypic ratio is 1:1:1 ratio. Now, Mendel work published in 1865, but it remained unrecognized till 1900 because of the lack of communication, because of the concept of gene factors, because it was not that clear that time. The mathematical approach for biology was not accepted, and no proof for existence of factors. So it got unrecognized till 1900. Now, moving to chromosomal theory of inheritance, which was proposed by Sutton and Bovary. They work out the chromosome movement during meiosis and the movement of behavior of chromosome, which was studied as parallel to the behavior of genes. So the knowledge of chromosomal segregation with Mendelian principles is called the chromosomal theory of inheritance. So according to this theory, chromosome and genes are present in pairs in diploid cells, and the homologous chromosomes separate during gamete formation, that is meiosis, and the fertilization restores the chromosome number to diploid condition. That means if the mother is having diploid number of chromosome and the father is also having diploid number of chromosome, they will be segregating their chromosome. They'll be like assorting their chromosome. That means the gametes will be having n number of chromosomes there, the gametes. Okay, when they fuse, the progeny or the zygote will be again having the diploid number of chromosomes. Yeah, it is shown in this diagram. Now, moving to chromosomal theory of inheritance continuation. So, Thomas Hunt Morgan and his colleagues conducted an experiment verification of chromosomal theory of inheritance. And this experiment was done with tiny fruit flies, it is Drosophila melanogaster. Now, why? Because it is suitable for genetic studies. It grow on simple synthetic medium in the laboratory, and they complete their life cycle in about 2 weeks. It is very important. So it is similar to the pea plant. It is easy to kind of complete the experiment and find out the results soon. And a single mating could produce a large number of progeny files. This is also very useful or advantageous. Now, clear differentiation of male and female flies. And also many types of hereditary variations can be seen with low power microscopes. So moving to sex determination, so we have XXXO type, which is seen in grasshopper, that is insects. So here the females are having XX genotype and the males are having XO genotype. Now, in case of mammals, they are having is like additionally to the autosomes, it is 22 pairs of autosomes plus one pair of the sex chromosome. So if it is XX, it represents the female, and if it is XY, it is male. So male is heterogamety and female is homogamety. Now moving to the next type of sex determination, it is in birds. It is ZZ and ZW type. So here the male is having ZZ sex chromosome genotype, whereas in female it is ZW female genotype. That is, in female it is heterogamety and male it is homogamety. Now mutation. So we know that mutation is the change in the gene or DNA sequence, and it causes change in the characters. It is phenotypic variation will occur due to mutation. It could be deletion, insertion, duplication, or addition of particular gene sequence. And this can result in alteration of chromosomes or like abnormalities or aberrations. It is chromosomal aberrations. Now, the mutation takes place due to changes in single base pair of DNA, and it is called gene mutation or point mutation. For example, sickle cell anemia. Now, coming to frame shift mutation, the deletions or insertion of base pairs of DNA is called the frame shift mutation. Now, coming to pedigree analysis, we know that the square box will be representing male, the round circle represents female, and this will be representing sex unspecified. And this is the affected individuals, this is mating, this is a mating between relatives, and this is the parents above and children below. And here this is the parents with male child affected with disease, and this is 5 unaffected offspring. Now moving to genetic disorder, there are Mendelian disorder as well as chromosomal disorder. Mendelian disorder consists of like examples like hemophilia, color blindness, cystic fibrosis, sickle cell anemia, phenylketonuria, thalassemia, and these are because of the alteration or mutation in single gene. Okay, it could be like a sex-linked, that is X-linked recessive disorder where the gene is transmitted from a carrier female to male progeny. Hemophilia is also a sex-linked disorder